So this time it seems to work, but I still wait until I see myself on YouTube, just to be on the safe side. Okay, it seems to work. I have to mute myself. So good morning, United States, and good afternoon, Europe, and wherever else you are in the world. Welcome to today's FIND seminar. My name is Carsten Schradi, and I'm a researcher at the CNRS in Strasbourg, France. And I would first like to look back what the talk we had last week by Wolfgang Goldman about competing females and carrying males on the ecology and physiology of reversal in sex roles. A very interesting talk on life history, behavioral ecology, and also physiology, which in the meantime, of course, is also available on YouTube. I had some problems live streaming last week. These problems obviously have been solved. And next week, we will have a talk by Noah Pinter-Wallman on spatial and ecological features that affect animal movements and interactions. So this is what last week happened, a very interesting talk, and next week will be also quite fascinating. And before I come to today's speaker, I would like to use again to make a short advertisement for a paper that was just published online today that has some relations to the FIND seminar, especially to the talk Eduardo gave in the second or third FIND. We had it all about the importance of not overinterpreting the p-value, moving biological anthropology research beyond p smaller 0.05. It's a very interesting discussion, and I hope we will have maybe one roundtable one time at the FIND to discuss these issues. And Eduardo will share the link to this interesting paper in the chat. I think he already did share it, if I see that correctly. But today's talk is very exciting. I know many people are very much looking forward by Hannah Coco from the University of Zurich, Switzerland, who will tell us about good reasons to live shorter lives. Hannah did her PhD in 1997 in Helsinki, and afterwards did a few postdocs, first in Cambridge and Glasgow, before becoming a lecturer in Finland, a professor in Helsinki in 2004. Moving to Canberra, Australia in 2010, and finally coming to the University of Zurich in Switzerland, where she is since 2014. She's famous to many people for her modeling approach in all aspects of evolution and social behavior, including topics like inclusive fitness, eusociality, lifespan, life history parameters, sexual dimorphism, selfish behavior, but also other aspects like disease, cancer. For example, I'm having a 2-day course at the University of Zurich where students discuss her modeling about cancer and lifespan and these interactions. She is since 2014 a member of the Australian Academy of Sciences, and she has just been elected as the vice president of the Society for the Study of Evolution, the position she will take up next year. And she will also be on sabbatical, so she will really have time and also the motivation to move this society more forward. If you have any questions, you're welcome to ask this or recommendation in the discussion we will have after her talk. She's published more than 170 papers in all journals from Nature, Tree, It Seems Proceedings, Royal Society. London B is her home journal, a lot of papers she has in there. So she's really one of the most important people in our field, especially for the modeling approaches. And we are very much looking forward to her talk she's giving today. She's at the moment at a remote, nice location in the Swiss Alps in the mountains. But unfortunately, there's a snowstorm happening there, the first this year, or at least this winter. And we hope the internet connection will be stable, but that's also the reason why she has switched her video off to have a more stable connection with us. So if she disappears for a few seconds, so far she always came back, then please be patient. And we are very much looking forward to your talk now, Hannah.
Okay, thanks so much, Carsten. And also thanks so much for organizing this thing, in these pandemic times when it just sort of keeps going on. Feels like forever. It's very nice to have these interactions. And I've had very many nice chats today when we managed to get anything serious happened, but right now a bit invisible here. But that's okay, because it's really the slides that you should be focusing on. I about the fact that those things because it makes you potentially live longer. You would get but of course, if you it wouldn't be nice to far like this work here. This is the EOS in this beautiful archipelago of Europe, maybe on these nice coasts of Antarctica, where.
Okay, everyone, you see it's difficult, so we will see how it goes. We already discussed if this doesn't work, then we will go to plan B and have a discussion about, now if I say the importance of statistics, Eduardo is hitting on me again, but it would then be the start of the discussion on a roundtable on the importance of statistical methods. It's unfortunate that this happens today. We will see if she's coming back. I mean, for the people that are, most people that are not in the fine organizing committee, the member of the 3 host labs that's in Chattanooga, in Yale, in Columbia, and here in Strasbourg, we always have also individual meetings with our guests before. I mean, Hannah had 6 meetings or so already, and that all worked well. And now she's back, we will see.
We need a, I think that, I mean, she did tell us it wasn't happening in the morning, but she maybe she would just record her, because she did say that it was snowing quite a bit. So I'm thinking that if for the first time, we shouldn't really be, while she tries to reconnect, seriously considering a plan B.
Yes. I mean, I also see, I mean, I'm in the same climate zone. The storm got, it's not so bad here down at the Rhine Valley, but it got much worse compared to this morning.
I actually managed to get back, but I mean, if I really do get kicked out like every 20 seconds, I'm not sure about this. I mean, I can try for a bit, but yeah, it's just
quite frustrating. I mean, Hannah, you know the situation better than all of us. So you're saying that it's snowing heavily, and so what is? I think we need to defer to you, since you know how the weather looks like and how in the past it has interfered with your connection.
It's been bad in the last half an hour, and it's I mean it's a bit stupid, but maybe I'll try once more, and if I get kicked out, let's reschedule for a nicer weather day. I should have actually stayed in the city, but this has not usually happened. So let me try once more, okay?
Yeah, let's just try. If we see it doesn't work, then we make plan B.
Okay, I'll stop sharing for a sec. I'll get the proper slide up, and I guess you didn't really hear very much about what I was trying to say here. So I was trying to make you imagine how you were born in this beautiful archipelago of Europe, maybe in the Eocene, or maybe in these lush coastal forests of Arctic. And then, of course, if you really try to live forever, you would have to adapt to all the changes that, did you lose me again? All the changes that happen when the world is going through its climatic shifts. Or it might be that you're born even a little bit earlier, like you're one of these wonderful feathered small dinosaurs during that bad day on the planet when all the big dinosaurs went extinct. This is actually, I've stolen from wonderful Daniel Fields. I think this is the cover picture of Current Biology. And his work has shown that in order to survive such a bad day as a bird, the ones that survived were the ones that had relatively straight toes, whereas the ones that had curved toes, or claws, they were the ones who clearly lived in trees, so the habitat just disappeared. And my point here is that if you do all possible environmental risks, for relatively flat claws, straight claws are the thing to do. And of course, I'm not really a macro evolutionary biologist myself, but we can think about some shorter timescale problems. If you think about, whatever the pop songs were cool when you were a kid, if you try to sort of be popular them today, it ends up being a little bit of a niche activity. So life, of course, has a solution to these kind of problems. Life generally involves a reduction, so you don't really try even to keep one soma, one organism going on forever. Instead, you think about copies of you or copies.
Okay.
I think we have to. That's our job, making the rough decisions. It's not fair to her. There's 70 people here. There are YouTube followers. We've been waiting to listen to Hannah present for a long time. I think she deserves to have the proper connection so that everyone gets to enjoy her and listen to her well. That would be my suggestion. I don't.
Yes, I agree. Also, the weather broadcast is that it's getting worse, not better. At least here, and I mean, she's by if you go like the bird flies, she's less than 100 kilometers away from me, so it's not getting better there either. So I suggest we have to stop this talk. We will ask Hannah to come back another time, and maybe. For the people who are interested, you're welcome to join us. And I suggest we have a look at this paper that Eduardo just published. So there's some extra exposure for him. And because all these topics we discussed many times in the fine, and I think many people will be able to say something to this. And I don't know how to go through this. One possibility is that we go through it and Eduardo says what he thinks is important. And I read this paper today and I marked obviously what I thought is for me important. And of course, for Eduardo, I guess everything is important. But maybe, Eduardo, do you want to quickly summarize or let's first ask everyone. Let's first ask everyone if this is okay. But I mean, you're also, of course, free to leave whenever you want. But it's at least, I think there will be some people for whom it is an interesting alternative discussion we can have now.
And definitely, I mean, first disclaimer, I did not boycott Hannah's internet, even when the paper came out today in the morning. So this was not, it's not crafted by us. But it really takes us back to one of the first meetings now almost 2 years ago with fines. So I'm not planning on talking much. I'd rather have an opportunity. We've been saying that it'd be so nice to have opportunities for chit-chatting about different topics. And if this one gets us going, that's fine. If it takes us some other way, that's fine as well. So this is just a very brief commentary. We are together with my wife and colleague, Claudia Valegia. We're just trying to get the discipline of biological anthropology, but I think the same applies to primatology. The same applies to actually behavioral ecology or evolutionary biology. Some of you may know the work that Valentin Amrain out of Zurich has also been doing. And contrary to the way in which by mistake we continue summarizing the main point, this is not about stats. Or if it is about stats, it is really about rebalancing, repositioning the role that mathematical statistics should have in our science. That's why I am hoping that I can get more people interested in doing this. It is about to really understanding, like so many colleagues in so many disciplines have said, that statistical inference is only one small portion of the process of scientific inquiry. In fact, Hannah was telling me today in the morning that in her talk, she'll be talking about some of these, about the process by which we acquire knowledge. And so what we're doing here is that there's a huge literature. I mean, I invite you to just send me if there's literature against the idea that statistical significance is almost worthless for observational research, like most of us do most of the time. In observational research, taking a frequentist statistical significance approach to inference is almost meaningless. It is not about changing the value of P, it's about changing how we look at data and how we look at science. And so what we're saying here is just, again, nothing new. We're only jumping on the wagon of the experts who've been talking about this for 6, 7, 8 decades. It's remarkable that it hasn't died off. What we're saying is that we need to be looking at the data for what the data are, and that there are many, many ways in which we can make progress moving beyond a dichotomous, arbitrary, thoughtless cutoff of 0.05. So really interesting conversation, because what we hear from students and colleagues, okay, what do I do instead? I mean, the p-value for me has become this security blanket. When you can go and say, I don't know if this is important, but p is more than 0.05. That happens with the students. They give you the term paper or the research study, and they don't know if it is important or not what they found, but R says it is significant. So we're asking you to throw away that security blanket and take responsibility for making sense of your science. Take responsibility as the experts, as the ones who really know the system, as the ones who really wrote those questions and the study you designed, the study you executed. Why what you have found out, it's important in biological terms, in evolutionary terms, endocrinological, genetical. That has to come first, then supported by proper inference with all kinds of statistical tools that we may not be qualified to fully develop, and therefore we may find ourselves collaborating with statisticians the same way that we have no doubts in seeking collaboration for doing endocrinology, genetics, energetics. You choose it. Any topic you do, most of us seek collaboration. So that's really what the commentary is about. It was very well received by the reviewers. We were delighted to see that. We don't know how the community is going to receive it. But again, I invite you just to check the list of references. It is not us. I mean, we're just the messengers of something that's out there. And we're hoping it will get into biological anthropology, primatology, ethology, animal behavior. So that's all I'll say. I still look forward. Some of you have indicated that it would be fun to have a time when we can talk about all this. Nancy's, so it sounds like you're saying that just because something's statistically significant doesn't mean it's biological. Absolutely. I mean, it's pure. You can do it with Excel. You can get any p-value you want just by changing the sample size. And so anything can be statistically significant and absolutely irrelevant, or the other way around. I mean, for years, the cigarette lobby denied the effects of smoking on lung cancer because some of those studies, which had low statistical power and small sample sizes, did not reach statistical significance. The same happened with the use of estrogen applications with women, and now we know that women treated with estrogen for menopause have higher risks of developing breast cancer. The list is so long of cases where statistically significant results were led us in the wrong direction. A few years ago, Steven Ziliak, who is an economist, this has been brought up to the Supreme Court in the US, asking them to please stop using statistical significance to decide on cases where the lives of people are at stake. Our problem is that nobody suffers. In most of the research we do in this community, there are no consequences for the lives of people, for the health of people, of making the wrong decision. I can go about life making any kind of wrong conclusion about the effects of grooming on pair bonding in titty monkeys, and who cares? But when you start talking epidemiology, when you start talking a COVID vaccine, then you understand easily that the decision cannot be based on a p-value. It has to be based on the magnitude of the effect. And only you, the expert, can tell if this is a change in blood pressure, in employment rates, in salary payment. Is this an effect that as a society, can we live with it or not? R cannot answer that. Statistician who doesn't know about how you approach these topics in a society cannot answer that. It has to be us, the experts, saying, sorry, I don't care what P says, but if there's a 3% increase in death rates because of anything, I want to do something about it. I've attended talks where survival rates lower by 25% in primates were dismissed, were not considered relevant for further discussion because they were not statistically significant. A 25% difference in survival rates between the offspring of dominant and subordinate infants. Well, for very, very small percentages, we make decisions for humans that are much smaller than 25%. So anyway, enough. I'm just open to hear other people's thoughts. There are colleagues in the audience who I know feel the same way. Please jump in. Or if you disagree, even more, jump in.
I have a question. Clara B. Jones, retired social evolution. If you're going to take this approach to p-values, let's put Bayesian analysis aside for the moment. But if you're going to argue this for p-values, how do you determine whether or not your results occur simply by chance alone?
Couple of ways in which I would try to answer that. First, the p-value doesn't tell you anything about whether your results occur by chance or not. So that's one of the wrong misinterpretations of p-values, but that's probably more technical that we need to do. I think that part of the change, it's a huge change we need to do, is to accept uncertainty. It's quite interesting that we go for statistics for precision uncertainty, where statistics as a discipline is the discipline most specialized in quantifying uncertainty and probability. That's what statistics does. It quantifies uncertainty. Still, we go there for certainty to make, you just said it, whether it is or it's not. But very little things that we're interested in are black or white, yes or no, dichotomous. That's part of the change, that we need to stop imagining that we're going to find the silver bullet that tells us, does the vaccine work or not? Because really, as scientists, we know that the decision is not being made. There is no such thing as the vaccine works or not. Is, are we happy with approving a vaccine that has a chance of working 90% of the time? Or do we want it to get it to 95 or to 98? And even then, we don't really know. I mean, it worked with the people who received it before. We don't really know if it's going to work for you. So part of the, now this is in trying to answer your comment, Clara. So whether your results are by chance or not require that you have an understanding of the population from where you're drawing your sample, that most of the times in our community, we just don't know. And that's the uncertainty we have to accept. I always tell the students that nobody has a problem. When you feel sick, you go to the doctor and they only draw 3 milliliters of blood, right? Which is about 1% of your blood volume. Nobody has a problem with the sample size, right? Nobody has a problem with whether that blood sample is random or not, because we understand the system.
Sure, but in that case, you're measuring discrete elements of the blood. So you know you have a standardized range from healthy to unhealthy, or acceptable to not acceptable. And that range has been determined by a large sample size. So all the doctor has to do is to compare an N of 1 to that predetermined range, which is based on a much larger sample size from the population. But aside from that, related to that and to what you were saying, wouldn't we solve some of these problems if primatologists, for example, or animal behaviorists selected their sample randomly? Wouldn't we get a more reliable guesstimate of what is going on?
The issue, I don't know if Michael is still there, Michael Taborski. I use his paper on he has this wonderful 2000, is it 2008 or 2010 paper on issues on the size of the sampling behavior, Michael. But this is a wonderful paper that I strongly recommend. I give my students all the time the challenges we face as we try to determine how we're going to produce a sample. For most of us working in systems where we don't get to choose, or we're very constrained in our choice of what to sample. Yes, of course, it would be ideal if you could sample randomly. But let's not forget that most of us, when we go collect data in the field, we have not just one question, right? We're not just testing whether these cells in a Petri dish are going to respond to the vaccine, but we're collecting data for lots of different questions that have different considerations. And we, most of the times, are sampling, like you said, Clara, we're not sampling blood from a human being, we're sampling owl monkeys from a forest that I have never stepped foot in before. So how can I decide what's random when I have absolutely no understanding of the population? And that's part of the problem that, I mean, and that's where also in speaking, I'm not saying I'm a firm believer that we must rely on the expertise of statisticians. They are the ones giving us now tools to look at our data without assuming population parameters for populations that we don't know, right? We can now really use many more non-parametric approaches to our data. So the answer of yes, of course, it would be better to random sampling. Some things you can do. I keep telling my colleagues that instead of trying to just use all the data you have, could you I mean, actually, many of our colleagues are these days using kind of resampling strategies that allow to control some of the issues that you, Clara, are making, I think, implicit reference to. But I think that what's lying in your question is that we have to accept how little we know about the systems we study. We're approaching them like if we were sampling blood from a human being, and we're not. We have no clue. When I sampled 10 owl monkeys, which ones did I get? And there's nothing wrong with that. The mistake, in my opinion, what we're seeing in the commentary is that we have to be more humble. We have to be more candid with what is it that I can learn from my system? What is it that I feel I have made progress in understanding, as opposed to setting up our science in terms of there is an effect or there isn't. There is a difference or there isn't. That's where I think the change, there's room for making change. Again, please check the references. That was our main goal. Just give kind of an entry point of reading on these topics to realize that in disciplines where a frequentist approach is costing lives, people are taking action. But we have no reason to lower the standards of our scientific rigor because it doesn't cost lives. I think that I've always told my students that the standards of a scientific study should be the same. I mean, we should be aiming for the same high quality standards regardless of the implications of making a mistake. I see the chatting, but I'm awful. Oh, thanks, Mike. Thanks, Michael, for sharing. Wonderful paper.
So let me ask you one more critical question after I read your paper. You say because in primatology, anthropology, you can't reach large sample sizes is one reason why the p-value should not be over-evaluated. Are you not afraid that people might need this as an excuse not to reach for larger sample sizes than they can? They say, okay, Fernandez Duque said we don't need large sample sizes, so I just interpret the little data I have and I don't put more effort into increasing sample size?
No, I'm not saying that. Again, I think that definitely, please check Michael's paper, because what Michael was saying, and he has a question mark, so I'll let him answer. But I'm not saying that we should not try to increase the sample size, because by doing that, the larger our sample for the questions we're interested in, we may be able to reduce the variance on which allows us to reduce the negative effects of confounding factors we cannot control. But Mike speaks about that in the paper. Mike, better to have somebody else talk. Mike, go, and then we'll take Michael, and then we take Sasha or Sacha. Michael?
Yeah, I think, can you hear me? I think it is a difficult issue that we need to somehow separate the different details of the scientific process to come up with a fair judgment. I think I fully agree with Clara that it is necessary to have some kind of procedure to make clear that the estimate that I make has some reliability and can be repeatable. So we need statistics to really disentangle random effects from robust and repeatable results. So I think what Eduardo mainly criticizes is the arbitrary value of 5% of error rate that has become a sort of consensus over the last at least 5 decades or so in biological research, not only in biology, of course. And that is something that is, of course, everyone understands. It's arbitrary and it's stupid. Why do we say that something that has a 6% error probability is worthless, while something that has 4% error variability says something? Of course, this is stupid, and we have to get around this, I think. And as Clara already mentioned, Bayesian statistics is one way to really come around this problem. But I think the other issue that has been addressed here is the importance of sample size. And of course, sample size is usually constrained, and that is something that I addressed in this paper that Eduardo was referring to. That usually or very often in behavioral research, the constraints on sample size are more important than the choice of a sample size by which you would expect to find statistically significant effects due to your considerations of the effect size. So this is of concern, of course. I mean, we are working in behavioral biology. We are working with animals, and we do not have unlimited access. We need to weigh all the ethical considerations and also the, of course, economic or financial considerations and the access to data to really determine a sample size that is sort of a compromise between what we expect regarding the effect size that we want to demonstrate or investigate, and these constraints. And we need to be aware, and there I agree with Eduardo, that we need to be aware that the messages that we convey also after providing statistical details should be really always taken with a certain degree of skepticism, not only because they are sort of only marginally significant by our statistical analysis, but also because of for example, the different criteria that we were able to consider in our study. That is also something that I think in behavioral ecology, over quite a long time in the early phases of behavioral ecology, was very important, that we did experiments with a sort of subset of the different potential influences on a system. And when the predictions were met, proved by statistical analysis, then we were happy with confirming the predictions of the hypothesis that we wanted to test. And this, of course, is a bit short-sighted, because if we do not understand the system well enough, and that is the point of Eduardo, I think, then we can well end up with wrong conclusions from even statistically significant results. So I do agree with Eduardo that we do need a proper understanding of a system to actually ask the right kind of questions and to select the right kind of approach, the right kind of approach that will allow us to consider all the potential influential parameters when we do experiments.
Thank you, Michael. I just want to reiterate your opening remark. This movement that some of us hope will finally bring some change, in no way it's asking that we stop reporting p-values. In no way. If you check Ambrain's letter, it's listed in the paper, Ambrain's letter in Nature, that within a week had 800 people signing it. They say 3 times. What we're saying is report the full p-value. It is just one more numerical index that, depending on the nature of your research and your system, may provide more or less evidence in support of your ideas. So that's very important. Again, my personal view is that for observational research of the type that most of us do, the contribution to understanding the evidence we've got through a p-value is extremely limited. Now, before we go to Sasha, Bayesian stats are not going to be the solution if suddenly we make of the Bayes factor another thoughtless criterion to decide what kind of evidence we've got. No single index should tell us about the importance of the data we've collected in the context of the questions we have. That's where I go back to my first comment of repositioning, rebalancing what some authors have called statistical inference and compare it with scientific inference. I strongly recommend the paper we cite there by Hubbard, Hubbard, 2019, scientific inference vis-a-vis statistical inference. But let's hear from, do you say Sacha or Sasha Engelhardt?
Hi, Sacha. Yeah,
Sacha.
Let's say in French. Sacha. I'm a postdoc in Michael Tobolsky's lab, and I mainly study these days Nori rats. And well, that's the model system. But a lot of questions about cooperation. And I wanted to. The discussion is interesting, and one of the things that I've taken to doing is trying to interpret the results in a way that is biologically meaningful. So, for example, I'll try and report, the estimates and the p-value, the effect sizes, but I'll also put them in actual biological terms. So, for example, if the grandmother is alive, her daughters will give birth to 2 additional offspring than if she's dead. When for social learning, for example, when there are experienced individuals, naive individuals will learn 150 hours sooner than naive individuals without any experienced individuals. Things like that you can put I get, to me, to give a sense of this is biologically significant. 2 offspring or 150 hours earlier seems to me to be worth it, to be a nice way of representing the information. I'd like your thoughts on that. And secondly, regarding sample size, I was working with demography data from human population, where sometimes there's like almost 200 observations, 200 individuals, 2 million individuals in a data set. And sometimes I've run analyses with 100,000 offspring to see if they survive. And of course, the p-value is much more likely to become significant at that point. I was wondering if you have some kind of solution for that, if you think of anything, if there's a way of, let's say, taking into account that you have such a large sample size. So that's that's my comment, and that's my question.
I definitely that has been part of the crusade, trying to get ourselves. I mean, we're into this business because we enjoy and we want to understand behavior, not because of the stats. So, and we sometimes forget. I mean, Hannah had told me that she was going to talk about this, that in so much of what we do, we're doing it with too frequently an implicit, an unstated assumption that the differences, the effects we measure, have evolutionary consequences, right? It's fascinating, I think, that in every introductory course, most of us probably tell our students that evolution can have huge effects over long periods of time. But then when we go and do our little one-year study and we just find a small effect, we dismiss it. So, and of course, that's a matter of time scale. Now, how do you decide what's important? In this comment, because we were talking to our colleagues in biological anthropology, I think we make reference to things like, what does it mean if I measure a 5 picogram difference in some estimate of cortisol in a fecal sample from a monkey? We have no clue, right? Not to mention that we're measuring it in feces when we're trying to understand something about how cortisol in blood, if the proper receptors in the brain are functioning, is interfering with some physiological process that will then affect the care provided by the male. So and we're just saying, can we at the very least go through this process of understanding where our data fit in this big scheme of things that we're looking at? In terms of your question of how to reflect on sample size for humans, I think that we have no trouble. I think it comes easier to us to understand the points we're making when we're actually talking about humans. I imagine the Chinese government having looked at changes in birth rates as they implemented their policy, one child policy, and they had goals. They had drops in birth rates that were important to them regarding of stats. So I think you need to have the question. You need to know the question you're trying to answer. You need to be clear on the process you're trying to understand to be able to assess if a difference of this magnitude is something I should be acting on. COVID has given us so many wonderful examples to reflect on this, right? And I keep telling the students, I'm teaching a class on this now, how frequently do you see news about the effects of COVID and the benefits of the vaccine shaped around statistical significance? I mean, I could probably find that there are no statistical significant differences of all kinds around COVID pandemic that people would be up in arms if I were to ignore, because it may not be significant difference between 100,000 people and 105,000 people. I don't know. So I think you need to understand the question you have to reflect on how do I consider this finding or this result? Not in a vacuum, not in abstract. I want to hear from others.
Yeah, I would agree that a small effect could sometimes be very biologically significant, and sometimes a very large effect is also biologically significant. It depends on the literature. It depends on what has been found already. It really depends on the knowledge that we have. Sometimes if you can, I don't know, I guess an example would be like in cancer research. Sometimes if you can show that a treatment improves lifespan by 1 month or by 3 days, so that might, sometimes that's a huge effect that can tell you, okay, we need to move forward towards this. There may be something there, even though it's only 3 days. So, yeah. Thank you. Have a nice day.
I have a question more than anything else. I'm not sure that COVID or, for example, climate would be really good analogies to what we usually do in the field with behavioral studies, because with COVID or, for example, climate, we're dealing with dynamic physical models. With climate, for example, we can usually only predict ahead for about 3.5 or 4 days. And then the data change again. And that's true with COVID also. So I think it depends on what kind of model you're thinking about and what kind of research you're doing. Again, I don't see much analogy between a dynamic model and a descriptive work.
I can comment if anyone else. I mean, but I think that any living organism is as dynamic as climate. I mean, we cannot predict what my cells are going to be doing, not 3 days from now, 2 hours from now. So it's as dynamic. But what I think is fascinating is, Clara, I fully agree with you. You're saying it depends on the system. And that's exactly the point we're making. And now I'm quoting Michael. It is stupid to have an arbitrary value of 0.05 for anything. It doesn't depend. It's always going to be 0.05. And that magic number, and God forbid you go 0.06, that magic number is going to be the only criteria you're going to be using for whether you get published or not. You're absolutely right, Clara. It should not be like that because it depends. It depends on the system. Are we predicting whether we evacuate human beings, or are we predicting if the lemurs at the Duke Primate Center are going to get cold? So the decision, if you're using p-values, would be the same, whether to evacuate people because there's a hurricane or evacuate cattle. So are you going to make the decision based on P, or are you going to make the decision of how it matters for people, cattle, insects, or you choose? Yes, it depends. That's the whole idea. And the experts know the system. I keep using experts. It's something that I never do in class. Those who study the system know the system. And therefore, they are best positioned than any coding software to decide what to make of the findings. Nancy says, I have seen in some field papers, not with primates, that some have used 0.1 as the cutoff. Maybe this is not acceptable anymore. What some of us, again, please remember that I am no expert on this, even when I was publishing on this now 25 years ago. 0.05, 0.1, it doesn't matter. I think that what we shouldn't be doing is not looking at the data for the data, not understanding our question, not understanding the system, and what is it that we're trying to decide on. Some people are saying that really statistical significance for observational research with limited sample sizes where you have no experimental setup, it is not meant for the kind of research that most of us do. I mean, that goes, again, you can browse the literature. I mean, what we're doing is a mix of two approaches, hypothesis testing and statistical significance mixed in this horrible hybrid. But that's more technical than I feel comfortable describing or explaining. Karsten, Nancy, hold on, let me see what Nancy says.
I think Michael has a question, has a comment first before me.
It was an attempt to try to be more flexible. No, that's another. The p-value says nothing about your hypothesis. The p-value is only the probability of you getting the data you've got. That's what I'm saying. My suggestion is look at the very basic statements that the American Statistical Association has published, so that a first step is understanding the misconceptions we have about what P gives you, because it doesn't give you what most of us usually think it does. That's the first one. And then just expanding the scope of evidence you're going to consider to feel that you have made a substantial contribution to understanding the system you're focused on. Tony's pasting here. Right. Now, I know that sometimes you read this and you get all confused. But again, why, something else that I found myself telling colleagues, why is it that we have no problems in following the advice of endocrinologists and geneticists, and you choose the subdiscipline, but suddenly when it comes to stats, I mean, it wasn't me. I mean, this is out there. All kinds of statisticians are alerting us about the misuse of statistical significance. So why not also follow their advice the same way that we go to a forest ecologist if I'm going to measure food availability in the forest? Let's see, Tony.
Yeah, I mean, I think we should get, Michael is waiting now for some time.
Yeah, I just wanted to go back a little bit to the question of statistical versus biological significance. I think what we need to be clear about is that, of course, statistical significance does not say anything about effect size. I think we need to be sure, and this is, I think, sometimes something that people may misunderstand, that a statistically significant effect does not mean that the effect is strong. It just means that it is repeatable. That if you do an experiment in exactly the same conditions, you will, with high likelihood, find the same result. But still, I mean, the effect may be minute. So, and this is, of course, also borne out by what Eduardo referred to when you increase sample size. I mean, with the enormous sample sizes that people get in epidemiology, for example, you get everything statistically significant, of course, because even minutest effects turn out to be repeatable. Yeah. So I think we really, when judging the value of a result and then the interpretation of the result, we always need to be careful about separating these two things. Is it repeatable, and does the statistical analysis propose that it is a repeatable effect? But is the effect important? Is it biologically significant, or is it so small that perhaps it still does not really explain important questions or give answers to important questions in biology? I just wanted to make sure that these two things should be separated from each other. And then I think it's also easier to judge the value of statistical inference.
Please, I'm all in favor of statistical inference, right? Absolutely. I mean, we do it all the time. I no doubt, because most of us are interested in understanding general patterns. If we're going to try to understand evolutionary processes, we want to be sure that what we're learning in this tiny little system can be generalized, is applicable to other systems, other situations, other contexts. So I'm all in favor of statistical inference, just to put it out there. The other problem with the frequentist approach is that it's very much the way that it's been traditionally done. It usually constrains us to just one hypothesis. I mean, it doesn't need to, but the truth is that when you take that approach, you end up considering one hypothesis at a time, when the truth is that we would all agree that for most phenomena we study, there are most likely several different possible explanations for that phenomenon. Which again, that takes us to maybe there are other ways in which we can try to make inferences from multiple models, etc.
Yeah, but now, Eduardo, you're opening a can of worms, because I mean, what you're addressing here now is the reductionist approach, actually, not just the statistical analysis. Of course, I think I'm very much in favor of a reductionist approach, even though I fully agree with you that it will end up with detailed answers on something that perhaps needs a much larger view. So I think we need to accept that we can only move forward in small steps by reductionist measures, by the fact that we sort of disentangle everything so that you can be investigated in ideal case experimentally. Yeah, and I think the failure that is often made is that we make much too large inferences from these detailed analysis. I think that is the major problem. And I find this all the time when I review papers for journals, that because of the competition for space in scientific journals and for attention among scientists, we are all sort of trying to make big inferences from small results. And this is a danger, of course, yeah. And I think this is part of the problem that, if I understand Eduardo correctly, that he's addressing. Even if we approach problems with the reductionist method, we should be aware of the fact that to understand the full picture, to understand the mechanisms that are behind a certain difference that we may find, need to be investigated in many steps, and only if we understand both evolutionary and mechanistic causation, we can really end up with a sort of comprehensive understanding of something. But I mean, this is all a bit trivial, because I mean, we as scientists, we know that we can only proceed in very small steps. And only if you're a layman and you don't know the procedure, then you may perhaps draw much too large inferences from small results.
I like what you just said, Michael, and it made me think that, yeah, of course, I'm also in favor of a reductionist approach. The truth is that what we need is we need everything. I mean, because you really want to have this mix where after you have really decomposed and broken into pieces the system to go inside the black box and understand the mechanisms, then you may find yourself exploring, well, the extent to which the mechanism is resilient or how it's influenced by other factors. So there's no single study. I mean, what you're saying, I agree. There's no single study which is going to allow you to understand the whole system. You break it in pieces, then you put it together to see how it works. So yes, absolutely. And actually the vaccine again gives us an example. Imagine, I mean, nobody would have occurred to say, okay, we need to start developing a vaccine. And if you were to choose a random sample of people from the world to test the first round of vaccine, well, you don't want that. I mean, you want to have a very well-defined study, which is what you, Michael, are saying. And let's take the baby step of understanding if the vaccine works in this petri dish with cloned mice, right? Because now we can really understand how the vaccine is working. But once you know it's working, you kind of start shaking the system and making it more complicated so that at some point you can decide, yes, I'm going to give it to all kinds of people all over the world. So I think that the process speaks of how there's value in all steps, but you really need to have the dialogue among them all.
I'd like to say a final thing. I'm talking too much. I would defer to Michael on just about any issue, even if I had to take that issue to my desk and think about it for a long time. But I would like to point out to students and to those who, like primatologists, work in subfields that argue that they cannot do field experiments. I think that Michael brought up the idea of experiments, but one can use math models and simulations to test the assumptions of one's study and also to conduct experiments with simulations. So I think that those approaches should be used more commonly.
So maybe I have the opportunity to say two things that are a bit mixed up before maybe we move to the Bayesian statistics of Anthony. I think we all agree that the dichotomist thinking below 5%, it's all important, and above, it's totally worthless. We agree on this. But I mean, there has been a reason why this has been introduced. And the reason was that people have some way to say, do we accept it or not? And now replacing the p-value by 10 different values and what leads to some social sciences where everybody can always argue in a way that his or her ideas are supported by the data that are collected, and that will make it very difficult to evaluate what is really important and what is not. So, I mean, there was a value of the p-value and why it was introduced. Overestimating it is bad. But the other problem that it produced is that often, even though we talk about 5%, people modified their statistics, they modified their questions, they modified which data to include into the analysis, that even in the past, in reality, many people were not testing for 5%, but for a totally unknown p-value, which they kind of manipulated to get down to this level. So there was a reason why it had been introduced, and just getting rid of it, it will make it very philosophically that everybody can always argue his or her way why the data support his or her opinion, and that there I see quite some risk in the revolutionizing of going somewhere else.
We haven't heard much from students today. You are the ones who always come say, well, but what do I do if I don't have the p-value? I'm not going to get published.
I mean, that's the other problem. We don't do science to improve knowledge, but most people are scientists, PhD students, postdocs that do not have a permanent position and that are forced to survive in this system, not by creating knowledge, but by creating papers that get attention.
But I think the students working with me can testify to how you can make progress on these topics, and you need to be prepared to debate with your reviewers and with your editors. But don't we teach that that's what science is all about? Exchange of ideas and trying to articulate your arguments why it's this way or the other. So you need to be ready for that. And what I mean, for those of you who are just getting started in the business of publishing, what I keep telling all the students is that you can always just say, listen, I'm following this literature. These are not my ideas, but if a reviewer challenges you, if an editor challenges, at the very least, have them go and read what you're citing. I mean, I'm doing this following this person. And so at the very least, the reviewer will have to explain why he or she disagrees with recommendations by the largest statistical association in the world, for example. So we usually tend to defer to the published peer-reviewed opinions of experts on any topic you choose. So read and cite, and that can give you some support to adopting a certain strategy with regards to reporting your data. But you can still do what they want you to do, but then expand. I mean, Sacha was talking about, make sure you have a section where you discuss what you think the results mean for the evolution of this trait or the other, for the physiology of this taxa or that taxa. Discuss your limitations. We always talk with Carsten about the business of publishing the high impact journals. Yes, they want you to present it like, if this is the end of the, I have figured it out, right? This is the paper that brings closure to the topic of the evolution of monogamy. Well, that's a decision you have to decide if you want to do or not, but there's no need to present your work like case closed. I figured it out. Karsten, have you been able to hear back from Hannah or not at all?
Yeah, she just said that that she has no connection with Zoom, and she hopes she can then present another time.
I'm sure yeah, what a shame. But I think we did. I'm glad we made that decision. It wasn't worth having anything less than high quality presentation today.
Eduardo, I just want to follow up on something you said. We're talking about biologically meaningful effects here and effect sizes, but I don't think anyone said the word study organism. I think you have to think in the context of your study organism too. You know, in my PhD work, I saw small differences in body mass in the offspring during early development, and that was due to communal care of the mothers. It was associated with communal care of the mothers, and argued that that small biological effect was important in the context of the rodent. Now, in an elephant, it probably doesn't matter, but you know, in a rodent, it does. And so I think that's another thing to have students think about is their study organism and what biologically meaningful effects means for that taxon. What are your thoughts on that?
Oh, I agree. I wonder if I've never done this, but I'm wondering if one could have access to all the data sets of Peter and Rosemary Grant measuring the beaks of the finches. And if we were to take just one study, and they found that over the course of a year, the average beak size increased whatever much it did. I mean, the data must be there. And I'm sure that, I'm not sure. I'll speculate that maybe it was not statistically significant because at the beginning they only had a few birds, or maybe because I cannot imagine beak size is going to change by 50%, right, in one generation. But suddenly now, 40 years later, we know that, you know what, those changes were kind of important. Now we have a whole new species with a new type of beak. We need to learn to live with the fact that we know so little about our study organisms. I have a paper where I reported that the only, and again, you look at my papers, my goodness, I mean, I've been guilty as everyone else, right, of doing all this. But there's a paper where I was looking at the effect sizes, the differences between males and females in some morphological traits. We captured our monkeys, and so we measure the kind of typical physical measures you can do in the field with the monkey. You measure the leg, you measure the arm, blah, blah, blah. And the only measurement that came out statistically significant was the length of the femur. And you're talking roughly 50 males, 50 females, all adults. And it was about 0.5 centimeter. And I just reported the data. I reported the effect size, and I have no clue what it means. Because as a person who knows about primates, I would have a hard time. I mean, I don't know. I would have to sit down and say, okay, how could a difference, if this were for real, if this is just something that is happening, that females have a 0.5 centimeter longer femur, or the femur, just to give a sense, the femurs are probably 20 centimeters long. Is there a reason? I mean, can I come up with a plausible process, mechanism? Can I come up with an evolutionary hypothesis where I can postulate that there may be some adaptive benefit to a female having a 0.5 centimeter longer femur than a male? So we report the data and hope that maybe, who knows, maybe 30 years from now we'll have an answer, maybe we won't.
I mean, for the biological significance, I have to say, I wonder why would anybody test for something which he does not a priori believe to be biologically significant. I mean, I myself have an example of a paper with a PhD student on alloparental care in striped mice, and we measured corticosterone and there was no effect. But the referee asked to test for this and this and this interaction. And I said, okay, if he asks and we test this until we find something significant, and so we can publish it. But in the end, for me, the paper was only biologically meaningful to show the amount of alloparental Karen striped mice and this relationship we found with an interaction with corticosterone. I don't even remember, and I would never cite this paper for this effect, even though we were kind of forced to find it and to put it in. So we have one more significant result, but I by myself would never have tested for that.
I just typed in response to that, Karsten, it made me think of, I think, Michael, you were alluding to this when you were saying that we have one idea, we go collect data, that one of the ways in which we can make sure, I mean, for me, so much of this comes to, do I feel like we have made progress, regardless of what the reviewers think? I mean, the easiest part, I mean, the young colleagues have a hard time believing that you'll get published. I mean, if you want, you'll convince them of anything you want. But at some point in your life, I hope you'll feel like, I just want to know if I know. I just want to know if I have really understood the system better. And Michael was, I think, Michael, you were talking about this. Andrew Gelman, who is, right, he's a statistician in Columbia. He has a couple of, he writes, he has a very interesting blog, I think, but also he writes articles for general audiences. And he talks about what he calls the researcher degrees of freedom. And that's what I think, Karsten, you were in a way touching on. All the decisions that we allow ourselves to make in the process, starting on when we start with the questions. And one way in which we can increase our sense of confidence in what we got is to be clear on the questions early on. Not start finding questions that fit the data once the data have been collected. So he has some very interesting readings on all the degrees of freedom that we never report, we're never thinking about, all the decisions we're making in the process of collecting data, curating data, selecting data. No, I'm not just talking p-hacking, abusive p-hacking like the one we've all read about. Much more subtle, that we're all doing without even being aware we're doing it.
Okay, I think we could discuss for ages, but we might also have to come to an end. It's also getting less and less people. I would like to ask if there's somebody of the people that didn't talk so much yet want to have the last word to say what they learned about it or what they are now thinking about, like maybe Alba or David or Birgit. Anybody volunteering there to make a short finishing statement, what you think was now interesting, important for you or to consider in the future? Alba?
Okay, this is kind of a trap. Thank you very much, Dr. Schroeder. Well, I'm Eduardo's student, so I'm kind of like aligned with his thinking about this. It is true that we kind of face this difficult path of, as postdoctoral or doctoral students, of getting published without getting in the trap of using p-values in a dichotomous way or something like that. Sometimes reviewers ask us, okay, but where is your p-value? This happened to me like a few months ago. I tried to do my inferences just based on the biological importance of my organisms. And at the end, I had to include the p-value because that's why the journal requested. So my advice would be like, find an advisor that helped you going through that path of knowing your system and knowing how to do your analysis and how to think about the interpretation you do about it. Is that a good summary?
Okay, thank you. I think that was a very nice final word for the live stream, which I'm going to stop now, of course.